So there are two major categories of methods in this imaging-based single cell transcriptomics area. And one category is multiplexed FISH, and the other category is in situ sequencing. Now, in the multiplexed FISH part, there are methods that use combinatorial imaging, combinatorial labeling and imaging to allow a large number of RNAs to be imaged in a relatively small number of imaging rounds, including, for example, MERFISH and SeqFISH. And I'll just use MERFISH as an example to tell you how it works. You know, there we assign air-robust barcodes to different RNA species, and we use encoding hybridization probes to target specific genes, specific RNAs, to imprint the barcode on each RNA, and then to read out the barcodes one bit at a time or multiple bits at a time by multicolor imaging. And then this is a targeted approach, means you select a number of genes to first before you imaging. But the number that can be imaged, we already demonstrated 1,000 genes in our first MERFISH paper, and last year we demonstrated 10,000 genes. So with the gene number increasing, it actually is getting closer and closer to be an untargeted approach. And then these genes can be detected with very high detection efficiency, close to 100%. And of course, one can also do more genes, more than 10,000 genes. And one of the questions is, what do you trade off when you do more genes? That'll be maybe a good thing to discuss. And then, so as I said, targeted may not be a major limitation here, but it's still worthwhile because there are untargeted approaches that we can compare with and then talk about pros and cons. And then for practical applications, there also may not be the need to image 10,000 or more genes. And very often we found 1,000 or hundreds of genes to be enough. And there's also within this category, another approach, which is by straight sequential imaging. For example, you do sequential hybridization and imaging. For example, you do 3-color imaging at a time for 3 genes, and then with 10 rounds, you could image 30 genes. Now, although this measures fewer genes, it's often very useful for very highly expressed genes where single molecules cannot be discernible. So, so also a very useful approach. And in the sequential sequencing, in situ sequencing category, there are actually both untargeted approaches and targeted approaches. Untargeted approaches such as FASEQ, and there, of course, the advantage is not targeted, and also it has a potentially single nucleotide resolution, which is another advantage. Compared to targeted approaches, including both targeted multiplex FISH approach and targeted in situ sequencing approaches, the detection efficiency of untargeted approaches currently is much lower. The targeted approaches include, for example, in situ sequencing and START map and other approaches. And this also have, for example, START map demonstrated the ability to image 1,000 genes. And as I mentioned, compared to untargeted sequencing, it has substantially higher detection efficiency. And so there are, you know, maybe various advantages and disadvantages of these different approaches. And I won't use the introductory time to go through that. If people find that to be an interesting problem and then areas where, you know, solutions can arise, we can discuss that. So overall, just to summarize, these imaging-based single-cell transcriptomics approaches, with these approaches, we can do gene expression profiling of individual cells in situ. And in situ is a strong point here. And that allows us to actually identify cells, their cell type and identity of individual cells in situ, and therefore directly map out the spatial organization of these cells with single-cell resolution. And even subcellular resolution of where transcripts are, and which is also important for many biological problems. So the spatial resolution is a distinct advantage of this, these imaging-based approaches compared to, for example, other spatial transcriptomics approaches, the ones that you hear in subtopic 3, the ones by spatially resolved RNA capture followed by sequencing. There are other advantages too if you're interested in those discussion. we can discuss that during this breakout. However, it is worthwhile mentioning that these imaging-based approaches do require currently specialized imaging setups, where the spatially resolved RNA capture followed by sequencing requires sequence instruments that is already widely available. So how to make these imaging-based approaches more widely deployable is perhaps another interesting topic that we can discuss, and many other topics. So I don't want to dominate here. So that's all the introduction that I want to do. And then with that, we can just start raising interesting problems that are both important, and also perhaps this consortium can find ways and collaborations to solve. Okay, so with that, who would like to raise the first question?
So I certainly have a question that I could offer in here. So thank you, Xiaowei, for that really nice introduction and summary of the field. And thank you, everybody. I'm really excited to be here, so I just wanted to say that. So one thing I think is interesting is, how do we balance the advantages and disadvantages of targeted versus untargeted methods? So are there good strategies or developing strategies or new approaches that will allow us to leverage untargeted approaches, say single-cell RNA sequencing, to help drive these targeted panels? Are there disadvantages to approaches like this that we should be aware of, and alternative strategies that could be developed? I'm curious if anyone has thoughts on this point. I certainly have a bunch of thoughts, but.
So this is Deborah. I was going to just be an observer, but I guess I'm now an active participant. As you brought up, as you think about other methodologies, I think one thing that's important is, of the current technologies, what are the QC metrics? What are the false negatives, false positives, to give us a good baseline of what do we have in hand and how good is it? And that should help to drive whether additional technologies or improvements are essential as opposed to necessary, or essential, or are the current technologies going to be good enough for at least the first round of inquiries? And how might we set up standards so that everyone in different consortia can use the same standards to allow ease in terms of interoperability of the different data sets that would be generated?
So I suppose in this session, the first session, we're primarily coming up with questions, right? So then if you have questions that follow up these two questions by Jeff or Deborah, then you can bring that up. Or if you just have new questions, you can bring that up too.
So this is Ed Lean. So I think an issue that confronts lots of us working with human tissues are problems with high degrees of autofluorescence. And this is really problematic for many of these image-based, fluorescence-based methods. And I think that it's also something that perhaps there might be progress in other organs before the one that you're working on. And it would be great to find some way of sort of collating and communicating advances and being able to deal with those kinds of technical issues across the different organs. In the brain, we have been dealing with lipofuscin and thinking that this was an isolated problem, but working with others have realized that it's a huge problem in the lung and other organs as well. So there could be some really generalized solutions to these things that could help lots of people.
Yeah, so I think I can follow up on that. Not only the fluorescence background, when we're moving away from model organisms that can be handled relatively easily and in a rapid way in a nice RNase-free environment for collecting the tissue. When we work on other, for example, human tissues and other animals and so on, the RNA integrity is another important problem. and how do we preserve the RNA integrity and then document the RNA integrity, and so that this can also help this kind of, that of course is not a specific problem for the imaging-based approaches, for all RNA-based approaches, but nonetheless, it is especially for human tissue samples, that is an important problem. And then for some particular organs such as pancreas, it's also a, whichever animal that you're using, that's also a important problem. So overall, not everything is as clean as a mouse brain when it comes to the tissue preservation for the RNA-based measurements.
I'd like to build on what Xiaowei just brought up, and I think this touches on Deborah's point as well, is that I think it's important to, in addition to establishing protocols that preserve RNA integrity, also have a battery of tests or quality metrics that can report on the integrity of the tissue. For example, the RNA integrity number, RIN number, would be a very convenient place to start. But in addition, what we need to develop is a rule of thumb or an intuition that links RNA integrity numbers to the performance of these different image-based techniques and how that performance might be degraded or potential artifacts that might be introduced. Because some of these methods may actually be able to work with relatively low RNA integrity number samples. And we'd like to understand what the relationship is between RIN numbers or alternative or better metrics of RNA integrity and the performance of these different image-based techniques.
Maybe I could bring up another challenge in this area. So we're all thinking about trying to make atlases and trying to do this sort of very systematically. In particular, as you go to non-mouse systems, you have problems of scale. The size of the tissues are enormous compared to what typically is sort of imaged with these types of methods. So how are we ever going to get a whole large organ coverage with these kinds of methods? I think is a big challenge. Otherwise, you were constantly looking at tiny little bits of an organ and lose the overall spatial context.
Yeah, I think that's a very good question. That's also in my mind as a question is, it actually is, I raise it as, is it important to extend to higher throughput? And of course, it's important to extend to higher throughput. The question is how. And then throughput, in terms of the number of cells, I think that's what Ed is primarily talking about. But we also talked about gene numbers. Is it important to increase the gene numbers? And the way I look at it is actually, it's always possible to measure more genes. At this point, it is not, there's no real technical issue of doing not only 10,000 genes, but also 20,000 genes and more. Then there's a trade-off when, when you measure more genes, you will be slower in terms of the number of cells that you can measure in a certain period of time. And then how to balance there. And then of course, the overall thing is not to have this overall figure of merit. For example, the combined, the gene number versus the cell number is fixed. It would be better to, overall improve the speed. And then what is the efficient way to improve the speed so that at a decent number of genes, we can actually measure with a very high throughput. And it is, as Ed mentioned, an important problem because you can easily calculate that for mouse, it's 1 centimeter cubed. But then for human brain, we're talking about brain, it's 1 centimeter cubed. For human brain, it's 3 orders of magnitude higher. So there, being able to measure a larger number of cells, substantially larger number of cells within the same period of time is truly essential. And I do think that for the Human Cell Atlas project is a very important thing to develop.
I have two follow-up points on that as well. I think this issue of the disparity between what we can image now and the size of the tissues that we're proposing to tackle is a really important one to address. And I think there are two aspects here that might be worth considering as well. The first would be, can we develop statistical methods that allow us to know how to subsample some of these tissues? For example, we don't need to measure the whole liver to be confident that we've captured the important aspects of that. And understanding what scale and what sampling is necessary, I think, is an important question. And the second is that one of the things that I think is an exciting potential advantage of these image-based approaches is that we're really still scratching the surface of what's possible in terms of imaging throughput. And it's relatively straightforward to think about ways to collect tens of terabytes of image data a day across really gigantic tissue samples. And that poses huge computational challenges and data storage challenges and distribution challenges. And so how do we, as a community, mobilize the resources we need to develop efficient computational approaches, both hardware and software? How do we develop standards that allow us to somehow be able to coordinate when the data sets we collect could be hundreds of terabytes of raw image data, if not petabytes, when we're thinking about the type of tissue scales we want to work with? And can we leverage ideas that have been developed elsewhere to address these issues?
So I think, hi, this is Pavel. I think I should sort of be maybe an observer because I'm not really an expert in the labeling part of these techniques, but maybe a question to Xiaowei or to the group. How much can you extend from sort of the current 2D, or is it even possible to do labeling and imaging in 3D without having to have a 2D sections? Because that would make it a lot easier for collecting, tissue volumes rather than sort of putting back things from 2D section to 3D section to 3D volumes.
That's another very good question. I think there are multiple reasons to think about doing it in 3D. And first of all, the penetration of probes, in thinking about doing 3D feels quite feasible. And as a matter of fact, in the StarMap paper that Carl Dijssel's lab published, they already have 3D imaging. And I don't immediately remember the thickness of the tissue in terms of their 3D imaging, but at least it shows that these probes can penetrate the depths of tissue sufficiently to actually allow 3D imaging. And whether 3D imaging, it certainly has advantages in terms of registration, right? Because if you have, especially think about multimodality, such as for the brain, you measure the activity with calcium and so on, you do 3D 2-photon imaging, and then you want to register to the cell type. That is a significant advantage. Now, whether it makes it faster or not, no, not necessarily, because if you slice an image, then the probe penetration can happen almost instantaneously. So because of that, it's not necessarily the 3D will improve the speed, even though imaging-wise it might, but not even substantially, but labeling-wise, it might actually slow things down. It has its advantage, as I mentioned, in another area, and that is for registration of multimodality images. And I think it is an important area to further develop.
What would be sort of the cost trade-off? Do you think in the 2D sections it is in a way cheaper, or because for 3D you also need to do large volumes typically, and if we do antibody penetrations or other penetrations, we get higher concentrations. How much currently is the cost an issue on these methods?
I don't have an answer off the top of my head. Maybe we could also work that out in the second half, because I think it is an important area to go into in terms of further develop the 3D volumetric imaging capability. But I think you raised the right question in terms of, in order to penetrate and then measure in a still reasonable amount of time, do you need to increase the concentration of the probes in the solution? And if you do, does it become substantially more expensive, right? And another thing, sort of along the same direction, but also cover what Deborah had originally asked, is some of the QC metrics. So one of the advantage of this approach, not this approach, these category of imaging-based approaches is, they have, at least some of them have relatively high detection efficiency. As I said, close to 100% with the multiplexed FISH approaches. Now, detection efficiency could be one of the quality control metric that one document. But a lot of these also trade-off was speed. One could imagine designing encoding approaches to allow lower detection efficiency, but faster. So was that, if the speed is a important thing, then how do we sort of balance between this? And another thing is that all imaging-based approaches benefit or enjoy is the spatial resolution. As I said, these approaches have much higher spatial resolution than other approaches. And the spatial resolution is something that if we can lose to some extent, for example, if we use lower magnification objectives plus amplification approaches, then we can also improve the speed. So where is the balance of the spatial resolution versus overall cell throughput is another area to think about when we think about this very large scale cell atlas effort. So these are all things that sort of tying multiple questions together.
Maybe I could just pose a big question. What constitutes a necessary and sufficient atlas using the FISH methods for any organ?
Can you elaborate that? What do you mean by what constitutes necessary and sufficient?
Well, if let's say you're trying to make an atlas of the mouse brain, what does that actually mean? What kind of spatial and gene coverage do you need to call that an atlas that is going to stand the test of time or be useful in many downstream sort of applications?
Yeah,
so within the
go ahead.
Sorry.
No, Deborah, you go ahead.
Okay. So within the context of HubMap and other efforts, really it's the cellular level in the sense of being able to identify single cells. And of course, then it's always a little bit of a struggle when we think about metabolomics, when the resolution is only 50 microns, right? So basically you get a glob, but you can't see individual cells. So I think perhaps one way of addressing the question about resolution would be, what are the components that we need to be able to distinguish? And I would argue, I think for most atlases, what you want to be able to distinguish is the individual cell. Whether that's something that's just strictly aspirational, I don't know. I would also say that that for the data being generated for HubMap and for HCA, it appears that there'll be a lot of combining data sets to come up with a consensus, which is great. The question then becomes, at what do these efforts need to worry about looking at individual samples? And I'm thinking of in the case of a patient with a disease. So you have one biopsy. And at that point, then the false negatives and false positives for any technology becomes really important, because you don't have the ability to average things out. And whether or not that is anything which is of a concern currently for the efforts that we're talking about here.
I think that front, maybe what Ed was also talking about, especially for brain, it's quite important. Of course, we are all already going after the single cell resolution, right? So we do want to get each cell its identity. But you know, how many different you know, clusters that you're talking about, you know, how fine you want to get to the cell type identification, or even whether they are cell types or states. You know, if at a coarser level, you could image with fewer genes and determine that. And then if you want to get the very fine level of splitting, then you need more genes. So the gene coverage will eventually be determined by how fine we want the map to be in this hierarchical tree of a cell cluster dendrogram, right? So I don't think that the answer is known out there in terms of what the gene coverage is. But I also agree with Deborah that we do like to at least get the single cell resolution. It's just the gene coverage in order to get to how deep of an analysis of the cell cluster dendrogram. That one is have to build with experience to see how much is needed.
So it sounds like what's important then, if we think on a cell level, is that the structure that's put together will allow for further refinement of being able to subdivide cells at a subsequent time as you get more data. So you can go from epithelial cells to the individual types of epithelial cells, perhaps even eventually into cell state or disease or non-disease. So the structure needs to be flexible, that you can further subdivide cell types as more data comes in.
I think one thing that I've sort of learned from interacting with the HCA over the years is how different organs sort of approach this, different organ communities approach this. You know, for example, in the brain, you know, spatial position is incredibly important, and it can vary from one histological section to the next to be in a different functional subdivision. And so an atlas of the mouse brain, for example, might need almost every section of the mouse brain to cover, and a different gene set for different parts of that section. Whereas part of the lung, you might be able to capture most of the structure in any given thing that captures the tissue microstructure. But I think it's maybe not the same answer. It's clearly cellular resolution. It's clearly pretty high resolution. But if you think about where we are today and trying to generate a whole brain map, it's not so easy to see how we're going to get that right away.
Yeah, I think, I mean, if you think about the first in situ LN atlas, which still I think is an amazing achievement, the Z resolution is perfectly fine, right? I mean, there was an every 100 micron, and it really covered, and nobody would now, whatever, 10 years or how long it is, think that anything was missed in that direction. The XY resolution wasn't enough, and obviously the number of probes or something. So the trade-off will be, and I think that's where the 3D is not really going to be that useful, even though it would be kind of cool to do 3D. It is the way I think you could, you make a reference value, which is something that you can do fairly easily with a high resolution, and then you'll be taking in a 2D high XY multiple probes and registering them into in some Z subsample, the Z scheme that will be different for different organs. So for brain, you say every, mouse brain every 100 micron, human brain every millimeter or several millimeters. But the XY and then how many probes, that will be the critical part for defining an atlas that would be useful and stand the test of time, I think.
So I'd like to pose another question. If I look at the list of names of folks on this call or in this section, there's a lot of the experts on these methods are on the call. But how do we extend these methods to other groups were interested in doing this kind of work within their own lab or as a service or some other way.
Yeah, I think this is a pretty important question is, as I said, how to make this method more widely deployable to a larger group of people rather than just people with special expertise in imaging. So there's both the instrumentation front and the analysis front. And the analysis front these days, we often try to make the software all openware so that everybody can use. The instrumentation front, then I would think that both commercializing the technology so that people can purchase, push button machines, and also detailed protocols for people who can, follow this, and also, maybe for the HCA to actually organize hands-on, workshops to teach people in a variety of different locations so that more people can use. There are various things that we could actually discuss in the second part, and that might be something that is immediately useful, so that these approaches can be more broadly used by a larger community. So for people who are not speaking directly for the problem, could you please mute so that we don't hear the background?
I wanted to toss in a point there following up on both Ed's original question and then this line of discussion here about how easy it is to disseminate the technologies. I think the need of a definitive atlas is really greatly reduced if the technologies are spread broadly. And I think the idea being is that now everyone can essentially reconstruct the portion of the atlas with the genes that they care about for their question. But if that's the modality that things are working at, then we have this issue. Can we develop standards that allow us to best integrate these different efforts and these data sets to ask questions that none of the different labs were trying to ask directly? For example, are there computational methods that you can use to integrate these data? And are there experimental standards that we could include? For example, if you like, a set of core fiducial genes that everyone agrees to measure. It's a small contribution because of the multiplexing of these methods, but those then allow people to better computationally merge different data sets and maybe even serve as important quality controls to allow different groups in different locations studying the same tissue to really be able to judge what potential batch effects they may have, or local effects, or artifacts in their data. And the question would be, how do we go about developing those standards? How do we go about identifying these gene sets, if you like, fiducial gene sets, and helping disseminate that out to the community?
Yeah, very good point. So I would like to raise some, further development to see, we could discuss in the second part. People can see how necessary or how urgent or how important these further development is. So because we focus here on the transcriptomics approaches, the imaging-based single-cell transcriptomics, but very often you find that it is useful to combine the RNA profiling with other modalities, for example, proteins, and then epigenetic properties of the chromatin, modification markers, and the 3D genome structure, and for brain even, functional function activity assays such as, immediate early genes or calcium and so on. So how key or essential it is to develop multimodal imaging approaches so that multiple aspects or multiple different modalities of these imagings, such as protein epigenetic property, genome structure, function activity, can be combined with the transcriptome. And is it a important approach that we should be talking about? Oh, I also got a reminder that we only have 7 minutes left. So we have come up with a lot of interesting questions, and we have more time in the second half. And it's also not to say that in the second half, you thought of an important question we cannot raise. But let's try to see whether somebody else have additional problems that you would like this consortium to solve.
So Xiaowei, I feel like I'm speaking too much, but for the sake of bringing up some other topics here,
Okay.
I mean, what if we could move a little closer to the analysis side of this? You know, what might an atlas be useful for? I mean, one thing for using these methods is to really understand the spatial organization of these cell types so that you could find abnormalities in that distribution in disease. And so, you know, I think one value of these sort of systematically generated data sets are some approaches to learn those spatial statistics and then be able to map other data to it to find out when things have gone awry, if there's been proliferation of some cells or selective vulnerability of others, for example. And I think it would be really useful for this field to kind of move a little more in that direction. And I'm sure that every organ system has its own diseases that's trying to do something similar. But the algorithms might be something which are more generalizable.
Yeah.
But
Yeah,
I know there's some. Go ahead.
Sorry, go ahead. I was just going to chime in that I completely agree with this, and I feel that there are groups that are doing some excellent work on developing spatial transcriptomic statistical methods. But some of the simple questions, like can I assign a p-value for a difference in a distribution, are still not really solved problems. And so being able simply to say, do these two different samples have different distributions of gene expression or cell types, I think is still more a qualitative analysis now. So I completely agree. This is a place where there is some innovation that's needed.
Yeah, perhaps also waiting for more data, right? Because, you know, sometimes the statistical significance is easy to establish, but sample to sample variation is not something that is a statistical significance capture. And then if you especially get to Ed's question of if you have a aspect that's different from a normal sample, from a diseased sample, then it has to be bigger than a sample to sample variation you see in normal sample, right? So then enough measurements needs to be established to see what the normal range is, and then to establish the diseased, you know, aspects and so on. So these are all, yeah. Any other questions that people want to raise? We have 4 minutes left before the break.
Maybe I could just raise one last thing since no one else is raising things. I wanted to follow up on, I think Jeff and Xiaowei, you both hit on this a little bit. It would be really great to find some place to sort of collate the gene sets that are reliable for detecting different kinds of cells, so that they could be sort of used more systematically and broadly by a bigger community.
Yeah.
That's maybe a place where it's easier to standardize across groups than the methods themselves, is the things that we're calling the readout for different kinds of cells. You know, at that point, you could start to think of this as, you know, a form of pathology measurement that you could be comparing across sites. And I think we're kind of in that place between the single cell omics to predict genes and the work that's being done to use those and show which ones really work well. Some sort of a place to put those would be really useful.
Yeah.
So
That
Ed,
sounds
this
great.
kind of leads in also to thinking about having a common standardized nomenclature, not only within a tissue that each group uses to identify their cell clusters or just how they're identifying their cells, but also, based on the opening talk, to have a standard nomenclature for cell types across tissues ends up being extraordinarily important. And being able to make sure that HCA and HubMap and KPMP and all of these efforts are using the same terminology. And it also means that having the common terminology also means that we have the opportunity to build some ontologies that would again further help the abilities to analyze data. So I completely agree with you on let's all agree on what we're going to call things.
So in this part 2, I think we're supposed to come up with solutions to some of the problems that we raised in part 1. And since we have a task of populating at least one blue box, and then this blue box, I guess you've all heard during the plenary talk that is proposed for this group to collaborate working on something to solve actually one of the essential and perhaps time-sensitive questions that should be solved relatively soon. So, we could still discuss open-mindedly a lot of the different questions, but do keep in mind that we're asked to provide at least one concrete solutions to one of those. So was that in looking at the group and then in thinking that, because some of those are interesting technological questions that maybe individual groups can have individual innovations and to deal with and so on. But some of those may be a kind of a question that require more of a consortium type of collaborations. So the kind of questions that are more amenable to this consortium collaboration type of approach might be the ones that we want to focus more on. So I was thinking about the questions that are raised in the first half. Then I thought one such category of questions is, in terms of how to make the imaging-based single-cell transcriptomics approach more deployable to a wider community. I think that, because in my view, although these methods, there's still room for improvement on various aspects, but they are already can be directly applied to solve interesting cell atlas and other biological problems. But how to make the greater scientific community that can benefit from this category of approaches, which in my mind is very powerful and very useful. And but it's just how in your lab to help you establish this approach, right? So then one of the set of questions that we raised are along that direction. And, various things that we could think about along that is how to train people to get the kind of expertise to do this. With the help of the consortium, do workshops to allow people to get both the knowledge training, but also hands-on training, to come up with standard sets of genes or whatever kind of a measurement to establish the standards so that results from different labs could be compared, but not just comparison, more importantly, could be combined to generate a more comprehensive knowledge if we all included these standards in, so that merging of data sets would be facilitated by these standards. Those are perhaps the things that as a consortium, it's built for making, coming up with solutions for that. So that's my thoughts about if we were to elect one of those. But of course, I'd be happy to hear your thoughts on, as a group, what we can work together on.
Xiaowei, I was wondering if maybe since the group has gotten a little smaller, it might be worthwhile just to have half a minute for people to introduce themselves, because there are a few people here I haven't had a chance to meet yet.
Oh, that's a great idea. Yes, right. So let's start some introduction. So could you,
Yeah.
I will start. I'm Xiaowei. I'm a professor at Harvard and Howard Hughes Medical Institute investigator. And we're very interested in this area, both because we developed a single cell transcriptome imaging method, MERFISH, and also applied it to various cell atlas effort. And Jeff is actually a key force in the development of MERFISH. So I'll pass it to him next to
Thanks, Xiaowei.
okay.
Hi, everybody. So I'm Jeff Moffitt. I just started my own lab a year and a half ago at Boston Children's Hospital and Harvard Medical School. I did my postdoc in Xiaowei's lab, as she just mentioned, and it was just a huge pleasure to be part of the team that developed MERFISH. And that's what my lab is continuing to use to create atlases of a pretty broad range of tissues.
So we can go around. Ed, you're next on my screen.
Sure. I'm Ed Lean. I'm an investigator at the Allen. I was actually one of the leaders of the original Allen Brain Atlas, which seems so low-tech now, of a single gene in situ hybridization across the brain. I lead a lot of the efforts now on looking at cell types, and particularly in the human brain. This is one of the methods that I've been sort of trying to foster the development of these methods to apply to human brain. And also have tried to lead some consortium efforts through CZI as well to try to bring groups together to solve some of these technical problems and compare methods.
Norbert, you're next on my screen.
Hello, my name is Norbert Tavares. I am a program manager at the Chan Zuckerberg Initiative, working on the seed networks in support of the Human Cell Atlas.
And Parvel?
Hi, Pavel Austin. I'm at Cold Spring Harbor Labs, and I'm particularly interested in sort of spatial imaging and trying to bridge between different tissues and different species, from sort of protein-based imaging or staining to what is being discussed here in terms of the RNA and transcriptomics.
Alexandra?
Hello, Alexander Denadai from Belgium. I'm part of the Human Gut Cell Atlas Consortium. I'm a great enthusiast of spatial transcriptomics, this is why I'm here. But we're just starting to set up our pipeline for SIGFISH and MERFISH as well.
Ilan?
Hi, I'm Ilan, also from Harvard. I work in Niels Gellenborg's lab. I'm a software engineer. So I'm working on general fluorescence imaging rendering on the web, which is sort of applicable to what you guys were saying about sharing data. So we have like a web-based renderer for high bit depth fluorescence imaging, these 16-bit images. So yeah, so that's what I'm working on now and developing tooling around that. We have the rendering working. So that's why I'm here, is to hear more about the sort of, I guess, biological side of things.
Tommaso?
Hello, everybody. My name is Tommaso Biancalani. I'm a research scientist at Broad in Viv's lab. I'm a computational person, and I'm also the moderator of the method section of this session here, which is in the afternoon, so the spatial profiling method group. So I look forward to seeing you there.
Sami?
Hi, I'm Sami Farhi. I direct the optical profiling platform at the Broad, and our goal is to take advanced imaging methodologies such as spatial transcriptomics, imaging-based spatial transcriptomics, try to make it available to people as much as we can, and in parallel combine it with functional profiling tools such as optical electrophysiology.
Steve?
Hi, I'm Steve Sansom. I'm from the University of Oxford in the UK, and my group's interested in inflammatory arthritis, for which we're using at the moment mainly single cell RNA-seq, but we're very interested in putting the cells and the context in the tissue, and particularly in spatial methods that have the sensitivity to detect genes that are relevant for inflammation, like cytokine transcription.
Kristine, did I pronounce that right?
Hi, I'm a postdoc in Tata's lab at Duke University. I'm starting to get started. I just started a couple of months ago and I'm getting into these techniques.
Heather?
Hi, my name is Heather Ann Brauer. I'm at NanoString, and I lead a lot of our consortium work and spatial genomics development work. And so I'm really interested in hearing some of the challenges that you all are facing and thinking about ways to improve upon that.
Deborah?
Hi, I'm sorry I missed the very beginning. It looks like we're introducing each other.
Yeah, because we thought Jeff suggested that if we all know what each others are doing, that we could probably, because this is a solution part of this breakout.
So thank you. I'm actually on the West Coast, and so I was up early this morning. Anyways, Deborah Hoshizaki. I'm a program officer at NIH-HCA. I'm one of the project scientists overseeing the data visualization core for KPMP. Also involved in HubMap, and I'm the officer for Rebuild a Kidney and GoodMap, which is a molecular atlas for the developing kidney and the lower urinary tract, primarily in mouse, but we're just now starting to get human data, or we were getting human data, because of course this is looking at development, and at this point we can't get any further human fetal data, fetal tissue. My main focus is that I just worry about a lot of stuff. I don't necessarily have solutions, but I worry about how things are going to be put together in a larger context, and I guess that's who I am.
Mm-hmm. Right. So I think that's everybody, and well, now we can discuss what would be the deliverable that we come out of this session at the end. As I said, I've made a proposal for those of you who have joined later, based on the questions, problems we raised earlier, that since some of them can be done by individual labs doing innovation, but then some of them seem to be very well fitted for a consortium effort. And especially given this, many people have raised this issue of how do we make the methods widely available to different labs and so on. So that has to do with both training the lab to get up to speed with the expertise, and then maybe we can have consortium supported workshops for that. And then also come up with standards so that different labs, when they measure, they know whether their measurement it has the right quality. And then also importantly, as Jeff suggested and Ad suggested, we have a large scale effort that's needed for this human cell atlas. And it's likely going to be done by contribution from multiple labs. And then if you collate all these different contributions together, it'd be good to have standards and references, standard set of genes or standard set of measurements that can be done at different places, so that when we combine data, we can use these references and standards to combine. So I thought that might be an interesting, but that's actually just to throw out my own idea of what the consortium might be helpful. But if you think other problems that are good for this consortium setting, we could discuss those too. But in the end, we should come up with at least one blue box, if you have heard from the plenary. And then I don't think it's very encouraged if we come up with 5 blue boxes. so it needs to have some focus there, although it's not limited to just one. Within that one could have related questions, but unrelated questions we cannot put in one blue box. That's what I was told.
So it seems like we're dealing with 2 issues. One is technology transfer, and the other is QC. And within that context, you know, how do you scale that up relative to two-dimensional versus three-dimensional in the scale aspects? Is that kind of a fair summary of
I think
kind
it's
of the
more than
big line items?
QC. I think the standard is more than QC because the standard could actually help putting multiple data sets together for a more comprehensive picture of the large cell atlas. So I think the standard reference does more than just the QC. But then of course it requires careful thoughts of what these standards and references kind of a measurement should be in order to sort of satisfy that goal, so that if you include those in your own measurement, then your measurements can be combined by, you know, with somebody else's measurement that also included those.
That's thank you for clarifying that. On the other topic about technology transfer, I like the idea of workshops. I would say in addition to workshops, a real low-hanging fruit is what we do within Rebuild Kidney, is that it's very simple. You can get $2,000, up to $2,000 to send your postdoc to somebody else's lab. All they need to do is they just email me what lab do they want to go to, 2 sentences on what they want to do, and an agreement that they will present a poster at our next face-to-face meeting. And so in terms of technology transfer, you can do it on a larger scale via a workshop, but I think there's something really valuable of also being able to allow postdocs to go to different labs and to get individually trained. It's something that perhaps is not really scalable. However, it does mean that we're setting up the next generation of collaborations by moving postdocs and fellows from lab to lab. And if we keep the bar really low, you know, 2 sentences on what you're going to do, you know, whatever the technology is, an agreement to present, and up to $2,000. It doesn't include supplies or anything like that. That's travel and a hotel. You got to feed yourself. But that seems to work well, both in terms of technology transfer on an individual basis and in building the next generation of collaborations.
Yeah, that's a good idea. If it doesn't overwhelm a lab, and then I think that is a good thing to do. While the workshop hopefully can handle more people. And these workshops should be designed in a way that there's not just a bunch of talks. It should be actually have hands-on session. The hands-on session, both in terms of a wet sample preparation and instrument, hopefully instrument construction and instrument using, and the data analysis. So that after the workshop, you could actually go back and start some experiments.
Just quickly following up on that point.
Thank you. Sorry.
Sorry, Deborah. This was just going to be a question for you, actually. I had a little bit of a difficult time hearing you in the last. In addition to NIH support for embedding postdocs, are there straightforward avenues to get support for the type of workshop that Xiaowei just mentioned?
So there are a couple of things. So the embedding the postdocs is what I do within the Rebuild a Kidney. And so we have some opportunity pool money that we utilize. And the trick is make it a reasonable amount. If you want to go to Australia where Melissa Little is located, then we bump that up to 5,000 just because obviously the cost of going from the States to Australia. But the trick is to make the bar extraordinarily low because, you know, folks are doing this because they want to learn the science. In terms of workshops, I would recommend that you can, once a workshop is established, that one can ask for travel money for young folks. You may want to pair up at Cold Spring Harbor or Mount Desert Island, a great place to be able to do both the tutorials and the hands-on. And again, getting the young folks there means that you're again building the next generation of collaborations. But definitely, you know, as much as, you know, New York, well, forget New York City these days, but as much as you may want to do this at a university, there's something about embedding people where basically there are no other distractions. A Silomar would be great, but they don't have any lab facilities.
Yeah, one thing that I'm just thinking, there are various methods. We actually do not have good representation here of people who are doing in situ sequencing approaches. It's more of multiplex FISH approaches. And Jeff's daughter just walked in. So it's interesting for these Zoom meetings. So the reason I mentioned that is, the workshops, I want to just remind people that in the category of imaging-based single-cell transcriptomics, there are multiple approaches, right? So if the workshop is to think about a overall workshop that cover all these approaches, then I think doing it in a place like Cold Spring Harbor is a very good idea. If we think about, even further lowering the barrier to make things potentially happen faster, could be, some individual labs with the help of the consortium bear the sort of responsibility of training people. Then, for example, Jeff and I, we could, set up a workshop here in the Boston area. And we could, for example, even think about collaboration with the Broad Institute to set this up, or some other things. But with my experience of setting up these kind of workshops, as I said, the most important part is the hands-on part. So because of that, we do want to have, previously when I do this for the STORM workshop, we actually prepare a lot of materials and buy a lot of antibodies and, reagents and make the cells, and then all prepared up front so that the people who come in can actually use these reagents, do sample preparation, image their own made sample, the sample made by themselves, and so on. So I suspect that traveling is something that for people who are very interested in learning, they might be able to cover themselves. But for people who are actually preparing these workshops, so there are real material costs and so on. And this probably would be helpful to have NIH or the HCA consortium to cover. Now that can happen for example, Mats Nielsen could train people on his in situ imaging approach. And well, that might be more easily happening in Sweden, and Ed Lang and Stan Lunderson could train people on the awesome, FISH approach, or the sequential FISH approach. And I don't want to volunteer for people, so let me actually just stop there because I don't want to name the names and then they feel like they're obligated to actually to train. I'm just using this as an example because we've done this before, and this substantially lowered the barrier because as a couple of labs put together, like Jeff and I, we could actually just organize this and then make things happen sooner. That could be one form. And then other than that, then there could be a combined form like what you said. It's actually fun to do, to see whether we can get the multiplexed FISH and the in situ sequencing people to all together to organize a hands-on workshop, whether it's at Cold Spring Harbor or whether it's at Woods Hole. These are the kind of things that could help training a larger set of people, both these individual methods workshop and then the combined spatial transcriptomics. If we want to be ambitious, we not only include the imaging based, we could also include the RNA capture and then spatially resolved capture and sequencing approach, like the spatial transcriptomics, like seq, and so on. So those would be bigger workshops and takes a little bit more planning and longer time to implement. But I think both would be very helpful for training the community on how to do experiments on this front.
So one thing I've one criticism I might have is that these workshops maximum say 25 people in there. So if we get 25 postdocs or grad students, then we've trained 25 labs for the year, which is not going to disseminate this very rapidly. But if we focus, say, on imaging core directors or genomics core directors or individuals from core facilities, it will facilitate more rapid dissemination rather than individual trainees from individual labs.
So I would say at this point, we should be saying and. So I think that your point's well taken, and eventually you do want the course to be well versed and capable of doing that as a core facility. Working backwards a little bit, as you're talking about training folks in a given technology, we're making the presumption that the technology is ready for prime time. And an additional workshop could very well be taking these imaging modalities and really, look under the hood. Tell us the things that are keeping you up at night about what you worry about this technology. What are the false positive and false negative rates? What are the issues they have? Can you use certain types of sample preps? Really kind of getting into the nitty gritty of what are the limitations of each technology. And I think if we could air all of that laundry, it then means that people have a better idea of when a particular technology might be better for their particular application. And if we know what the dirty laundry is, and then thinking about which of the technologies are really good enough to go forward to mass utilization, that gets you then into the workshops that you want to do. And it could be, from individual labs. If there's enough mobility with that, it could be then moving into also teaching the cores how to do this, and then also trying to get folks moving from lab to lab. And I think there's great value in doing all three of those. So what I'm seeing is kind of 4 different flavors of workshop, which all I think add something unique to the whole technology advancement and building the next generation of the consortium.
I think one of the things that we also should keep in mind is that this family, the imaging transcriptomics family of spatial methods, I think is the most dynamic. There's the versions of MERFISH that are coming out, they come out every couple of months, which is amazing, but it also means that it's very difficult to, I'm in some sense a core facility director, and people will come and ask me, okay, so which version of MERFISH should I use? And the fact is, I don't know what to tell them. The answer is they need to try one and then probably try the next and then modify for the question. My sense is that,
and it also depends on what tissue you're using and what kind of resolution that you need. And is it, are you okay with a lack of resolution but increase in sensitivity? So yeah, all of those things.
And there's 2 things that I think there's a thing that you can do on an organizational point of view in terms of dissemination. And I think there's something that we can do on a collaborative basis, which would also be helpful. From an organizational point of view, I think it's helpful to target labs which have both the resources and the will to spend significant amounts of time getting this set up and running. For other technologies, what I've seen is that usually there will be one lab from an institution which will reach out and say, we're willing to be the guinea pig for our institute. If we get it to work, here's a list of 5 or 10 labs that sort of trust us and would be willing to work from us. And I think if we can find such like lab-based power users, those would be the 25 different postdocs or grad students that I would target in the training session. The other thing that I would do, which we've been participating in the Tumor Atlas Network, and I've had a really great time with it, is to distribute a core set of samples which we think are well representative of real things that you might experience. So this is getting back to Jeff's point about RIN numbers and Ed's point about the different levels of autofluorescence in human tissues. If we could get a big block of human, I don't know, liver, kidney, brain, maybe at different age ranges, and set up a bank which can distribute these samples broadly to different labs performing these methods, so that you could put up a sample of what your technique can achieve in a particular tissue type. It would lend a lot of guidance to people as they're trying to decide what to ask next. And it would let them evaluate where their sample may fit on such, compared to these samples, compared to these standards.
Yeah, so in other words, not just coming up with standard gene sets and so on so that you could compare, but also come up with a standard set of samples so that various labs could measure. I think that's a good idea.
And honestly, I don't think it should be even a standard set. I think if possible, it should be the same sample, which is distributed. Of course, I don't know how to get that going from a compliance point of view, but hopefully we,
It's very doable. KPMP did exactly what you described. They got a chunk of nephrectomy tissue. It got divvied up amongst the 5 different tissue interrogation sites. And basically, you know, the idea is looking for biological consistency, in this case, over a whole variety of interrogation technologies, whether it's single cell, single nuclei, proteomics, or metabolomics. But that's exactly, I think, the right thing to do.
Yeah.
And it's not that hard to get a chunk of tissue. You know, that if we can't figure that out, then we're really
then we can't do anything.
So maybe I can mention one thing that we're doing in HCA. I'm part of a standards working group in HCA. Is trying to come up with a forum for compiling the versions of these methods that work in different tissues. At the moment, this is just for the single cell omics, and you know, compared to this field, that's a relatively standardized field, and yet every tissue there are different things you have to do. So they don't all work the same. Some are, once you try to isolate the cells, you've released digestive enzymes, and it destroys what you're trying to do. Others are easy. Some are very fatty, some are not. And so we're coming up with actually a publication, but then some sort of a way to sort of publicly disseminate much more broadly the sort of current state of the field, compiling what the experts are doing in each of their respective organs. And I could see something like this being extended to these types of things, where undoubtedly every tissue has its own different tweaks on how you have to process the tissue. You could extend it to the types of standard measurements you would want to do, like RINs on tissues, like a poly-A probe, like if someone comes up with ways of doing membrane labeling in combination, things like this that could hit a much bigger audience than a couple of workshops. I have to say I'm a little skeptical about using workshops as a way to really broadly hit a community without a big burden on the people that are running the workshops.
Yeah, but I think they solve different problems, right? So I agree with you that Ed, that you know, what you suggested is very good, and then I think it serves to tell people what these methods can do. But then there are people who say, oh, I love to use this method, but I don't know how. So that's the thing is, you know, in my mind, you know, commercialization is the, you know, eventually the way to go. But before commercial units are out there, there are people who are so eager to actually, you know, do these experiments, and they don't have the expertise to do that. And how do we help this group of people?
So I think, again, it's an you know, posting the protocols and really all the gory details. We did that in RBK for making organoids, and Melissa Little is a member of RBK. And, you know, there was a little bit of a pushback. Well, it's already been published. Well, what's published doesn't necessarily have all the additional tweaks that go on. And so RBK's website is completely open, and we get everyone to put their current protocols in with lots of details. And I think that that did a lot to drive the utilization of making organoids, where now it's become pretty standard across labs. And it may be that you know, organoids might be a little easier than trying to get the spatial context of our transcripts sorted out. But again, I think that having a central place with the detailed protocols is also a real positive.
Yeah, absolutely.
And yeah, and you know, and I think the point is, I think the point that you made about each of the things that we've proposed is fulfilling different needs.
Yeah.
And as you can tell from my point of view, is building the next generation is really important to the NIH.
I think everything that has been said is good. It all fit this one big blue box. So, I mean, it seems like we're all having a go of, you know, disseminate the technology so that a much wider community can use it. And that includes both standardization, benchmarking, detailed protocoling, and training. And then this training could include both training the core facility and train individual labs and so on. So I agree with Deborah that one should not exclude the other. These are all ands, right? So it's good that in this one big category, because if you go out, I do think there are people who have curious mind to say, oh, can you do this and can you do that? But then most of the questions that I get when I go out give talks is, how can we do this? And that is indeed somewhat special in this category, because as I said, you know, for example, if you think about spatial capture and sequencing, such as spatial transcriptomics and the slice-seq approach, then there people do benefit from the fact that in their institution, they do have a sequencer there, right? You know, it's these days it's very hard to find out an institution that does not have a high-end sequencing, you know, capability. And then they just get these slides and then they can get going. Now this one does require an instrumentation. And then this instrumentation, how do we get it out there? Eventually, I think a commercial instrumentation will get out there. But before commercial instrumentation get out there, how do we help? Then detailed protocol certainly is good for people like, you know, Ed and Sami. They can set up the instruments in Allen or Broad. But there are also people who, you know, back in my STORM days, I learned this. You know, they do. I always tell people STORM is very easy. You can just do it in no time. And then I learned that there are people who have not done imaging before, and then they don't agree with my statements. So I think for these imaging-based, whether it's multiplex FISH or in situ sequencing, because you do need a imaging setup there, and how can we help quickly people can gain that expertise to do this kind of measurements? You know, I think all the thoughts that we mentioned today in terms of dissemination, they're all good.
So on the question of protocols, I've been doing a little landscaping in this area, and one thing that has come up quite a bit are the protocols. So a lot more details from start to finish, particularly the fluidics when it comes to things like merfish and seqfish are really complicated and challenging. And the complaint I've heard from folks that already have an imaging background and have labs that can really handle this is that those protocols are just not detailed enough. Mm-hmm. And the protocol actually for merfish on protocols.io is one of the better ones. It's the best one actually, but it's still missing a lot of pieces in there that a lot of groups can't seem to fill in. So it's been on my mind to start making some calls mm-hmm. and asking folks to how we can improve those. But let's imagine I've made that call now to everyone mm-hmm. here. Hey, can you make a better diagrammed, blueprinted from start to finish, right to the analysis, what's going to be the pushback that I can expect to hear from you? And why haven't protocols that are already there been tailored to be complete to the point where another lab can replicate them or institute them?
I don't think you get major pushback. It's just time, right? So, you know, usually these things are done by postdocs, and they're all graduate students. They all are busy trying to finish their projects and graduate and get onto the job market and so on. But I do think these things should be done and could be done, you know, because when we help other places set up the capability, we not only share detailed protocol, I think we all take pictures. We also take pictures of what the fluidics actually look like. And then all these things can be made in a way to help people to actually do. It is a helpful thing to do, you know, because indeed a single workshop, you know, if you want to make it very hands-on, can only cover tens of, you know, labs or facilities. And then if people can use the protocol and then already attempt to setting up themselves, then it will be more broadly useful.
So one way of making sure you get detailed protocols is to have the backing of the funding agency. And so for us, you know, every meeting we have, every 6 months, anytime anyone gets up and talks, I require the first slide is, what have you posted up on the website? You know, what data sets have you posted? What protocols have you posted? What collaborations have you established within the consortium and outside the consortium? And that slide is one of my first metrics I use when I start looking at progress reports. And so if the push comes from on top, that you really need to get these protocols in, I think that would help.
I want to toss a point in here too, and I think this is a great conversation, and you know, especially you know, when I was in Xiaowei's lab, you know, speaking for her as well, I think we felt that it was very important to help get these protocols out there and be as clear as possible. But you know, when you write a protocol, there is an assumption of the people you are writing it to, right? Just as you sit down to write a molecular biology protocol, you're not going to walk through the details of how you teach someone PCR. There's a sense when you provide the protocols for building up custom instrumentation that it really is very challenging to teach someone, say, Python from the beginning. So we would put all our Python code there, we'd put everything there, we would try. And so I think these things come in waves, and the protocols are put out there to be complete for the people who have some of the skill set that can allow them to step in and do this. And we've been getting feedback, at least for the Merfish fluidics, that there are people who are having a lot of success with that. But that's not to say that there isn't a whole group of people that we need to see if there are ways that we can improve our protocols that, you know, require less of an upfront technical knowledge. And one thing I think could be useful along these lines is to help establish a user and community where these questions can be posted, say on something like Slack. And that also will lessen the burden on the early people who develop these technologies to answer some of these questions as well. I don't know if in the context of some of the workshops or efforts that you've helped push forward in the NIH, Deborah, if you've seen efforts along those lines, or if, you know, Norbert and CZI, you have things that are going that way as well. So the question is, are there good ways to establish an online community where others can help answer questions, not just the initial people who develop the protocols?
Yeah, put it on protocols.io. So protocols.io has a very active forum and discussion. So once the protocol is there and people try to use it, it's going to generate a set of questions, and from there the protocols can build into the questions. There is, within the HCA and CZI Science Slack, there are several different image-based spatial transcriptomics, you name it, chat groups that have various levels of activity depending on what's going on at the time. But I guess more can be done. Are you guys a part of the folks on this call, those communities? Join.
So protocols, the protocols IO is something that we've been really interested in. KPMP is using that, and I think that we'll probably, RBK and GoodMap will probably switch over rather than trying to maintain the protocols on the website. It's just nice to have a one-stop shop. And if people go to the website, then there'll be direct links going out to the protocols IO. But yeah, no, I think that's the way we probably should go.
So let me say this. So we're supposed to come up with some solutions that the people in this Zoom room, at least some of us, can collaborate on and working towards. So in this dissemination part, you know, how do a few groups can collaborate on this approach? Or the approaches that we just described, so that it is a collaborative effort in solving an important problem?
I am happy to on all sorts of things here. This is an area that I've been thinking about at CZI. So with the protocols and standards, that's something I'm happy to work with folks and sort out how we can do that. The workshops idea is something we've been thinking about, but I haven't made much, taken much action on that. So actually a question just for organization for our blue boxes here as well.
Yeah.
Would we divide this into standards as something separate from dissemination, training, that kind of thing? We
could, but they, in my mind, are somewhat related. I was told that related questions could all populate a single blue box. So, so dissemination standards, you know, I think these are, because the standards do help dissemination. And so I think they could, that's just a procedure thing. If we put it in a one blue box, it might just be easier to do. But, you know, we could put everything that we said so far there, because they all sound like good ideas. But I think one of the goals to say that how do the people who are participating in this discussion can collaborate and fulfill some of the goals. So I was just looking at how we could collaborate on these fronts.
Well, certainly I'm pretty happy to be involved in any workshops with the spatial, image-based spatial methods. You know, my lab has, I think the Murfish stuff that's up there on Protocols.io, I think was an early translation of some things that we wrote in Xiaowei's lab.
Yeah, I think it's some nice people who translated that and then actually asked us to check. I think we really appreciate their effort. Is this the CZI people who helped us translate our protocol into the Protocol.io?
Protocols.io themselves. So anyone who's a grantee for CZI can actually save themselves some work by having protocols.io folks actually do the translating.
Yeah, I think they did for that. And then we did check those. And so, yeah, so we could collaborate with, you know, CZI to do more of these detailed protocols. And then the standards, how do we come up with that?
Well, I think if there are multiple people that are adopting these image-based methods, then it's nice to have a couple places, you know, actual geographic locations that are running these approaches in developing these standards. So, you know, I think it's maybe a little bit unfortunate that there aren't people from the in situ sequencing side here, but certainly I'd be very happy to help, you know, to measure some standard samples, to help validate some standard samples, to help characterize, you know, fiducial or standard gene sets in a range of different samples. I think that's something that we'd be, in my lab, pretty happy to help with. I think we want to find other partners because we really want to make sure that these standards are deployable. And we want to have help in these efforts that when we devise QC metrics, that we really see that they perform consistently in other people's hands?
Yeah, I think that's exactly our, reason of being. So that, of course, we'd be very happy to help with that. I think we should think about the range of tissue types. I think that clearly the inventors have had the most experience with not just what works, but also what doesn't. I think it would inspire a lot of confidence if you say this is something that we haven't had good luck with. Put it out there and see if your method does better, or if it doesn't, then you know better. So we have, in terms of range, I think my official role within the HCA is on a kidney project. I'm happy to generate data in kidney, any protocols we work out there. And we're actually going to do hopefully Murfish and Codex as well. So we'll have a nice comparison between protein and RNA-based approaches.
Yeah, again, so the protocol part is actually somewhat easier. When it gets to standard measurements and workshops and so on, it seems like it needs more significant effort and perhaps also even needs support, right? So, the lab who bear the burden of, like Jeff just nicely sort of volunteer to do some of these work, but these work needs to have actually financial support to carry out such work. So I don't know whether this would come from NIH or come from HCA and things like that. This is a front that it is important, but it is not that easy just to do it on the side.
Yeah, no, it requires significant investment.
Right. So then in terms of doing these standard measurement to help the field to establish standards, I'm glad that I have representatives from two major institutes here, Broad and Allen. They're very good at, helping with the standard setting, but it is for individual lab to do that. They need support. Good that Deborah is back. We're saying that some of these things will require financial support to do. Protocol, we don't think we need financial support. We'll just diligently work on the protocol front. And the standard measurement, helping setting up standards and references to help to build these references for the bigger community to look up to help the dissemination and the product. Just like the workshops, even on this front, the labs who are doing that, it would be nice to think about whether there are financial support to help this kind of an effort.
Right. So I apologize for having left the call for a moment. Since we're sheltering in place, I got myself a puppy, and we're still doing puppy training. So given thinking about standards, it might be worth tasking individual organ groups to think about who has a chunk of tissue that can be shared amongst the technologies as a start, and at least, and then to build up to combinations across. I'm sorry, I've got to let a dog out.
I might want to put, while Deborah's taking care of her new puppy, I want to, when we talk about sharing tissues, it seems to me that perhaps, even though our goal is human tissues, it might make sense to establish a handful of mouse organs as the beginning, because then this really allows reproducible production of these tissues locally before we get into the challenge of shipping between labs and the natural variation that comes in clinical samples.
Yeah, I would agree with you, Jeff. I think there are so many extra layers of challenges with the human tissue that you want to take advantage of the model organisms to establish robust technologies and standards, and then adapt it so that you can work with the more challenging tissues. But it's not a good place to start on that sort of thing.
Yeah. So what might be helpful is if this group is going to stay together in terms of dealing with this, is to have Sanjay Jain. He's part of HCA. to either that or Jeff Spraggins to talk about what KPMP did to take one chunk of nephrectomy tissue and to ship it out, and all the it's just not a matter of shipping it out. It's, you know, what buffers and what conditions and all of those things. I think you're thinking in the background of, oh my God, this is, it's not just getting a chunk of tissue via FedEx. There's a lot of stuff that goes involved, and they may be able to give some insights because they've done that for kidney. And they've adapted for kidney biopsies. So they may have some lessons learned there.
This is Laura Barisoni. I agree with Deborah, but also I agree with the previous comment. It is challenging to work with the human tissue, and there are so many other layers of things that we need to think about and organize that perhaps moving not quite in parallel, but almost, but maybe test something on the animal and then immediately create a protocol that can be applicable to human, I think will move the science a little faster in a way, and place it in almost in real time into a meaningful context for clinical operation or clinical research at least. So maybe think about something that can be also pushed together at the same time. We test and then we implement in human. We test in animal, implement in human, et cetera, versus trying to do everything in animal first and then go to human.
Yeah, it sounds like each individual organ needs its own reference and standard, right? So a set of genes for kidney, a set of genes for liver, a set of genes for lung, and certainly we know a set of genes, you know, for brain, and, you know, for different organs that needs to have these measurements. It's actually, you know, quite a lot of work, but I think it's a good thing to have.
Do you think that we need to impose a recommendation for a set of genes? Or is it better to have a standard organ and to then say, here is a set of genes that work for me? It will give you a lot of knowledge about the background autofluorescence, the RNA integrity, and then if you want to try another set of genes, you're looking at, you're not trying both a new organ and a new set of genes, you're just trying a new set of genes in a familiar tissue, and you have a reasonable expectation for what might work or not.
But different set of genes for different individual labs of their interest, of course, but if we're coming up with something that people can look at to say whether they did this right and whether the experiment worked and so on, then have a reference list of genes that they can compare to, right? Sure. And then it gets to a bigger picture of, you know, when you do have a variety of different measurements, and then if you want to put it together, have some common sets there to help sort of align the measurements and then put it together and to generate a more complete picture. That's also good. So of course, what are the set of genes to measure? You know, maybe it takes actually some trial and error, you know, not only to do bioinformatics to look at, you know, what genes matter, and then also not just the matter biologically, but also matter technologically, you know, at what kind of, you know, abundance range and what kind of gene length range, you know, all these things, then that, you know, those are what matters when you set up a reference or standard for measurement for also technological, you know, reasons and so on. So I feel like it might not be easy to be able to right away come up with, but eventually to have such a set is a good thing. And then, you know, some labs might try to measure only a subset of that set of standards, but still that could help align the methodology, I mean, the measurement outputs together.
This is something I'm currently working on. Actually, I'm trying to set some reference genes in the gut, but one question that's still intriguing me is which number of genes are necessary to, let's say, define cell types and states, the minimum numbers. Has anyone any experience on this?
Oh, I think it depends on actually the complexity of the different number of subtypes of cells that you see in your the organ of interest, right? So if you get to the brain, you know, the number of gene requirement is not small because very often in a relatively small region of the brain, if you look at the neuronal subtypes and populations that you can generate, you found that to be on the order of 100. So with that, of course, you could imagine that the number of genes imaged would be relatively high. But if you find an organ that says that if you clustered for example, a single-cell RNA-seq data, and then you found 15 different clusters, then I think you would need a relatively small number of gene sets to do that. So I think it does, it's a moving target depending on the tissue and depending on the granularity that you actually want in your eventual cell atlas.
Yeah, and so for bladder and prostate, they're dealing with exactly the same issue. There's no one single gene that says, aha, this is, you know, a endocrine cell in the prostate, but rather it is a combinatorial. The cell expresses A, B, and C, and doesn't express F and G. So, and I think that as we look in more and more tissues, that this combinatorial identification is going to become more and more prevalent. Sorry, Ed.
No, that's okay. It's a good point. I mean, it's been clear from the single cell omics for a long time that we're all in a combinatorial space here. But also it is a somewhat of, at least many places there is a hierarchical organization at some level. And so you can hit sort of different levels of granularity with different sets of genes, with more or fewer genes. It's still very unclear in many cases what kind of level of granularity is meaningful for your biological question of interest. But I think I tried to make this point a little earlier, but I think, you know, not all probes work equally, not all predictions from single cell translate into something really clear cut with the spatial methods. Some place to sort of collate what gene sets work for what kinds of cells for each organ community would be really useful.
Mm-hmm.
Otherwise, we're all just reinventing the wheel constantly, and you can't compare to one another.
That's actually a really nice point too, because you know, there are all these customized probes being made all over the place for each of these technologies, with the exception of the untargeted in situ sequencing, and it could be quite useful to have a common repository of those sequences. I mean, I know we've got a pipeline in place, so we just basically regenerate these every time we need them. But for places that they've got experimental validation, there's real value to being able to put those someplace that's not, you know, buried in a supplement of every different paper that's published on this.
So we have, I just want to remind everybody, we have only until 1. So we have 10 minutes left. And I think it's an incredibly helpful discussion. You know, we have a lot that we can, you know, in this area that we can put into the blue box. And these all have to do with making the methods dissemination and making it more broadly deployable to the bigger scientific community for using, including training, including the lower hanging fruit is, you know, as detailed as possible, hopefully interactive protocol generation, so that, you know, not only we try to make it more detailed, but also hear feedback from the people who say which part is missing and then add those parts. And then to workshops, and workshops could be both putting all the spatial transcriptomic approaches together as a larger kind of a workshop in places like Cold Spring Harbor or Woods Hole, or individual, methods could have its own workshop, and then that probably easier to be done at the location where the experts who develop the methods can have those. And then to the point of standardized sample, a common set of sample for different organs that are sent to different labs for measurement that establish standards. And then also not just a common set of sample, but hopefully eventually come up with common set of genes that could be used not only to calibrate the methods, but also to combine measurements together into a more complete biological picture. What do I miss something? Okay, good. And we have 8 minutes. So any other good thoughts? They're all welcome.
So I would also include that the technology transfer is also what is it? Unforeseen consequence or foreseen consequence is also trying to build the next generation, because I think that's going to resonate. When asking for money from a NIH standpoint, that would resonate as opposed to just we want to do a workshop to disseminate this information.
You mean to build the next generation of collaborations? Or to train them?
The next generation of folks using the technology. It'll also be the next level of collaborations. I mean, we find that within RBK that although we're saying all the PIs should be collaborating, the PIs aren't in the lab. The people who really, the people in the trenches are all the postdocs and students. So if you really want labs to collaborate, you get the postdocs collaborating with each other.
Yeah, absolutely.
And that really builds then really meaningful collaborations, and again builds up for the next generation.
I can also guarantee you that if these are hands-on workshops, it will be postdocs and graduate students who's attending. It would not be the PIs who are attending the workshops. So
we will need the PIs and developers though to teach the workshop.
Usually the hands-on parts are also taught by the postdocs and graduate students in the PI's labs, and the PI comes in to give some lectures. But that's a great way to establish. We actually have established collaborations that way through workshops like that before. So, yeah. All right. Anything else?
So we don't really, I think, probably have time to go into this, but this, this conversation went very much in the direction of sharing methods and developing standards, which helps to kind of push the community and develop the next generation, as you say. But I also think there's the issue of how to maximize the value of what we're getting and pushing analysis methods. And, I think those are things which can also translate across this. I mean, the intention of this whole workshop here was to bring together all of these groups that are sharing common methods and have some common ideas of understanding this fine organization. But this is all methods and not the analysis and what you can learn from the data. And I think there's a lot of space for trying to foster some community development work on the algorithmic side. And I don't know if there are others here more that are more computational that want to comment on that.
Yeah, absolutely. So I think we should, include that even in this blue box of, develop next generation analysis approaches. But in the afternoon, there is actually designated. That's why in this morning session, we more put emphasis towards the experimental method part. In the afternoon, there's a spatial profiling analysis breakout session. And Tommaso is actually the moderator of that. Tommaso, you want to say more about your session? If he's still here. But the afternoon, just for those people who are interested in analysis, certainly go to the spatial profiling analysis. But I think here, it is an important thing to mention because indeed, not only the computational people are doing a lot of great work in terms of doing analysis development for spatial profiling approaches. The labs that are developing the spatial profiling approaches are also developing analysis. And to put together these analysis to sort of enhance the common understanding of what do we learn in terms of biology out of these measurements, different modalities, how to put them together, and so on. This is really an important, it is worthwhile. Do people have other thoughts on this front that we could enhance this part a little bit, even though we have only 3 minutes left?
I have a minor point on this. I mean, I think this is a great point, both that we need these new spatial methods, but as methods developers, we're often already involved in developing some of these methods. But one thing that I think could really help drive the development of new spatial analysis techniques are standards in the data output. We have so many techniques, and as Sami says, the versions are changing. And if we could standardize, there could be essentially a white paper on this is the format, these are the things that are there. And if that was carefully designed and adopted broadly, I think that could make a huge difference in how many people with a computational background choose to sit down and actually try to develop some of these techniques, and how easy it is to then test different techniques, analysis techniques on these range of data.
I think that standardization should be the priority in many ways, but also time-wise. And so your idea of having a white paper is actually great, because we can do a lot of things, but then we have to go back and rethink it because we haven't standardized it, or we haven't done it in a way that can be widely useful to everybody. And so it's something to think about sooner rather than later. And that is where I think we will have the most of the impact in the long run.
So I should say, there's a lot of effort going on in this, and who knows, probably each consortium has its own effort on this. But CZI has sponsored a lot of work on this with their Starfish program. The BRAIN Initiative has been working on standards which largely are sort of adopting some of those things from CZI. This just may not be well known, but I think actually this is a really excellent point for this kind of meeting, because we can understand what's happening across the different groups and try to coordinate them. There's no need to do this many times. I mean, I'm not sure what the best mode of dissemination for this kind of information is, but a lot of it is already ongoing.
Yeah, I agree. I mean, actually, it looks complicated, but actually, the spatial information and so on, it's not hard to standardize the output. In a way, with the BRAIN Initiative, we're already have a set of recommendation, right? And then, as Ed said, a lot of them did just come from the CZI. So it's something that is actually quite easy. And then we could just sort of come up with some documents of that, and then send out to different communities and so on.
So it sounds like one of the outputs from this meeting may be some centralized location of who's doing what or what, because I know about a number of these efforts for standardized output formats, but and I know about them because we tried to start some of them and discovered that there are others already doing them. So it'd be helpful to know who's doing what in what consortia so that we can join in or not duplicate efforts.
I would say try to get everybody in the same room. Honestly, that's probably the biggest output of this, because this is what happens. I think a lot of people appreciate standards. When we were putting together the first pipeline for analysis of MERFISH data, we thought about these standards and put them there. And standards are somewhat easy enough to put in place that everyone develops their own, and then we're in the same scenario. So now is a good time to get everybody in together and try to hash out exactly what those standards would be. And if they're already in place, it's even better. Just getting them out so people know to adopt them is great.